All right, so I'm excited to have Kelley Tilmon with us this morning. She's Associate Professor here in the OSU Department of Entomology, where she's the State Field Crops Entomologist. And Kelley's been involved with a multi-state study to look at soybeans as a plant that pollinators enjoy visiting. So she's going to talk about that study this morning, talking about pollinator diversity on Ohio soybeans. So Kelley, I'll turn the mic over to you.
Okay, well, thanks very much, Denise, and good morning to everybody. I really appreciate you taking the time out of your busy schedules to join us this morning for this webinar, and I also want to thank Denise for inviting me to present. So, as Denise said, my primary responsibility in Ohio is research and extension on field crops, which often means in Ohio corn and also soybean pictured here. And a lot of my work revolves around integrated pest management and insect ecology issues in soybean. And a lot of it boils down to education on how to best manage pests, including different approaches to pest management. I cannot claim to be an expert in pollinators or pollination, so why am I speaking to you today? Well, I would like to tell you about 3 studies that I have been involved in, which all involve some element of soybean and pollinators. The first study I'll talk about is specifically looking at the diversity of different pollinator species that we find in soybean. Briefly, I'll touch on a thing that's still really in play. We haven't finished this study, but a study looking at the time of day when honeybees visit soybeans. And I will touch on a couple of highlights from a study from a few years ago looking at neonicotinoid seed treatments in soybean and how that touches on pollinators. So a little bit about soybeans. This is a very important crop in the Midwest and in the U.S. As a whole as well. We have in Ohio, where I'm speaking, about 5 million acres of soybean, which is a lot. In the Midwest, approximately, we grow 67 million acres of soybean. And there's even more in the southern, there's additional soybean acreage in the southern states, particularly down through the Mississippi corridor. Now, soybeans are actually self-pollinating. They do not require pollinators to provide pollination service. So why are we talking about pollinators in soybean? Well, even though soybeans don't require pollinators, pollinators seem to like to visit soybean. We have some evidence, it's not strong evidence yet, but there is some preliminary evidence that pollinators, even though the soybean plants don't need them, that having a good pollinator community can actually enhance yield, which is very interesting for soybean farmers from a management of their fields perspective. If there's anything they can do to enhance yield, that's usually considered a good thing. And we also care about pollinators in soybean because if they are in fact visiting these soybean fields, then this is a potential source of good pollinator habitat. 67 million acres in the Midwest alone is a lot of potential good habitat for pollinators. And so we were interested in teasing this out a little bit more. So the first study that I'll talk about is specifically looking at the different pollinator species and the abundance and diversity of those pollinator species in soybean. So we're not just talking about honeybees, even though they are, one of our star pollinators, but many of the native bees that are also important pollinators. This study is funded by soybean farmers, specifically through commodity checkoff boards of Ohio Soybean Council, and also the North Central Soybean Research Program, which funds more regional projects using money contributed by farmers specifically to fund research in soybean. So This is a multi-state study where we looked at the pollinator species in Ohio and 6 other states in the Midwest. But I am today only going to focus on what we found in Ohio. So just a little bit about soybean growth stages to orient you. Soybeans in Ohio are typically planted in May, and then they go through several different growth stages, vegetative growth stages, until they reach about the midpoint of their development where they begin to flower. And R1, R refers to reproductive stage 1. That's when we first start to see a few flowers on the plant, and then the R2 stage is when the plant is in full flower. In the next 2 stages, R3 and then R4, which is not shown here, we start to get pod set and the beginnings of pod fill, although there may still be some flowers on some nodes of the plant. So we're really, from a pollinator perspective, we're really interested in this period here from R1 to approximately R3, where there may be flowers available for the pollinators. And typically, and this varies depending on planting date, plants are usually planted sometime in May, but there's a several-week spread when planting may occur, and the variety and maturity group of the soybean will also play into when flowering occurs, but typically the last half of July is when we see soybeans in flower in Ohio. So how did we conduct this study? Well, we did sampling in 8 different farmer soybean fields in 7 different Ohio counties. So we really tried to get a good coverage of Ohio. And this was really a collaborative effort from a lot of people, and I'm going to mention some of those people on the next slide. But basically, we collected samples of pollinators in a trapping device. We call it a bee bowl. And this is a cup that's painted really high-tech, really technical research equipment. A solo cup that we spray painted this attractive yellow color, mounted it on a fence post, tied to the fence post with zip ties. And this allows us to move as the soybeans grow, we can move the cup up over the course of the season so that it's always hovering right around canopy height. And in this cup, we put in water with a drop of soap to break the surface tension. Pollinators are hopefully attracted to the cup, fall into the water. And then we collect these water samples twice a week during the flowering and early pod set and early pod fill. And depending on the location, we were collecting samples from July 1 through August 25th for this study. These are the counties where we did our sampling. The furthest north was Ashtabula County, where we had 2 sites. And then we also had single sites in Darke, Fulton, Knox, Licking, Logan, and Pickaway counties. And this, I should mention, was a sampling that we conducted in the summer of 2016. And I really want to highlight the collaborative nature of this. First of all, these were all farmer fields with farmers who volunteered to have this sampling being conducted. And I was really, really impressed because when I put out a call for participants to provide sites for this study, I thought I would have to twist some arms to find some sampling locations. And actually I had so many people raising their hands and volunteering to be a sampling location that we couldn't do them all. We couldn't cover that much, that many sampling sites, and so we had to just kind of do a few. And so I was really impressed with the eagerness of the farmers to cooperate. Also, this was a cooperation with OSU Extension educators. These are the boots on the ground extension people located in the individual counties, and these were the folks who actually, once we went around and helped them set up at the beginning of the study, the folks in these counties were the ones who went out twice a week to collect the samples so that they can then send them to us. And so I want to give a shout out to these extension educators: John, Lee, Sam, Amanda, Mike, David, Eric, and then also my research associate, Amy Rodenbush, who helped a lot with this study, and Pat Bozay at North Dakota State University, and I will touch on his role in a moment. We also had these nice little signs that we put out in the fields where we had these sampling units. And if the farmer wanted them, if the farmer didn't want to draw attention to it, we didn't have a sign. But actually most of the farmers thought it was kind of cool to have a sign in their field showing that there was OSU research going on and that were interested in studying pollinators. So in each of these fields, we would have these pollinating sampling posts at various distances from the edge of the field and at varying distances into the field. So there was more than one sampling unit per field because we were also interested in how far into the field are we collecting which pollinator species. This is Pat Bose, an entomologist at North Dakota State University, and his role was really absolutely crucial. He is the fellow who identified to species level all of the species that we collected in not just Ohio but in all 7 states where this study was conducted. And that might sound like a simple thing, but actually when you're looking at all of these native pollinator species, it is something that requires a very high level of detail and skill to be able to identify these things to species level. I can't really overemphasize how time-consuming and how skillful that job is. And Pat had the duty to take thousands, literally thousands of specimens to identify and process for us, and he just did yeoman's duty on that. So big shout out to Pat Bose at North Dakota State University. So in Ohio, we collected a little bit over 2,600 bees in this study, and this is just the bees. We also collected a lot of different pollinating fly species, and we're still kind of organizing and going through all these samples. So I'm really focusing my comments on the bee species that we collected. And this was an extremely surprising result to me, that the incredible diversity of pollinating of bees that we found in Ohio soybeans. We found 49 species from 5 different families. I was expecting a couple families, maybe a dozen species, but to find 49 was really very exciting and to me surprising. This table that shows the details of how many bees from the different families is available in the report that Denise has made available to you. Also available in the report, which you can download from this presentation, or also you can access it at this link, are all of the different families and the different species and the numbers found. And I'm not going to dive into that in any detail, but if you are interested, you can go look at that in more detail. And I'm just going to hit a few of the highlights of these results. These were the families, 5 different bee families that we found in Ohio soybeans: Andrenidae, Apidae, Collectidae, Halictidae, and Megachilidae. And this was the most common single species that we found in the study, species called Lasioglossum hitchensi, which accounted for 36% of those 2,600 captures. So it was a very common species. This is in the family Halictidae. In general, the family Halictidae, which are the sweat bees, were the most common family in the study. So not just that one species, which comprised 36% of the captures, but 96% of all of our captures were in this bee family. So this was by far the most abundant bee family represented in this study. The other family, Apidae, this is the one that contains the honeybees and the bumblebees. They were represented. Collectidae, these are plasterer and miner bees. These are sort of lesser known bees. They are solitary, and they're called plasterer bees because they have a mouthpart secretion that they use to line their nests. And when it dries, it's sort of a cellophane-like material. So they've plastered the inside of their nest with this material. Andrenidae, also called mining bees. You can see, if you look carefully, one of these bees on the ground, and these are ground nesting bees. So here's an example of one of the nests that they might dig for themselves and their offspring. Megachilids, these are leafcutter and mason bees. These are familiar to a lot of people. And this is why they are called leafcutter bees. They cut these circular discs out of plants and use this material to help make their nests. So those are the 5 families that were represented, again, the majority being the Halictidae. Now this is some data on when we found these bees and when they were most abundant. So our sampling extended from the 1st of July through the 26th of August, depending on location. So we sampled in the flowering and the early pod set, pod fill stages, and that varied by geography and by the field as to when that was. So we didn't sample every field for this entire duration, but we were sampling at least some fields from this period. This axis here is the average number of bees caught on the different sampling dates, just summed, averaged across all of our sampling locations. And these are the different sampling dates. And the upshot of this graph, and we're just looking at the halictids and also the apidae, the halictids being the most abundant, 96%. So the upshot here is that really the bulk of our sampling success and indicating the bulk of the activity of the pollinators fell in this last half of July, which makes sense because it's the back half of July that's typically the peak flowering period in Ohio soybeans. But we were finding them really throughout the season. This is data on the distance of the sampling device from the edge of the field. So we had sampling positions at 0, that's the edge of the field, and at 5 meters, a meter being, more or less a yard, 10, 25, 50, 100 meters, and then however big the field was, we would go to the middle of the field. And the point of this was to see how far into the field we were finding the pollinators. Are they just focused on the edges? Are they going all the way in? And what we found, there's really not a lot of difference at all in the number of catches on how far into the field, showing that they're just going throughout the field more or less uniformly. And I think our furthest sampling point for a midfield was a very large field where the middle of the field was 250 meters from the edge. So that field was about 500 meters from side to side, which is half a kilometer. So very big field, and we were still finding pollinators in the middle. So the take-home points from this particular study is that Ohio's soybean fields are habitat for a very surprising and wide diversity of pollinator species, which is very exciting. And also it shows us that soybean farmers are providing really extensive habitat to support these pollinator populations and the ecosystem services that pollinators provide. So, moving on to the second study that I've been involved in. And this study is still ongoing. We were collecting data this summer, and in fact, we're still working up these data. But this is a study also funded by soybean farmers with funding for this study from the Ohio Soybean Council and the North Central Soybean Research Program. Looking at what time of day do honeybees visit soybean fields. So we were trying to make this manageable, so we were focusing on honeybees because they're big and easy to identify and easy to see. And this was a collaborative study with Dr. Reed Johnson, who is our honeybee biologist here at Ohio State University. And this is an important question because if in fact we do have so many pollinators in soybean, and if it's serving as good pollinator habitat, we want to be able to have good advice for farmers for if they want to focus on stewardship of those pollinators. What time of day are they most likely to encounter pollinators in their soybean? And in particular, different species may be foraging at different times. So this is just a preliminary look looking at one species of that being honeybees. And it's important to know when the pollinators are most active, because then if a farmer is making an insecticide spray decision, if the farmer is obliged to use an insecticide to manage an increasing pest population, and if he or she has a choice of the time of day when that happens, maybe he or she would choose to pick a time of day when the pollinators are less likely to be active. And so in order to get at this, I think all of us have a kind of an instinctive idea of when pollinators are out and about, but to really nail it down, you need data. So the way we conducted this study, Reed Johnson was kind enough to set up some beehives for us next to a soybean field on one of our research farms for Ohio State research farms. And then we put up sampling devices at various distances through the soybean field, similar to the other device I showed, but a little bit different. I'll show you the difference. And we only did this at one site this year. And not only did we check the bee bowl traps, but we also at the same time made visual scans of soybean foliage. So we would have a timed count of, I think it was like 3 minutes, where we would walk through the soybean field and record any honeybees that we saw. And this was a modified bee bowl where we had bowls of 3 different colors because they might have the color might have a different level of attractiveness for honeybees. And we did this every 2 hours from 7 a.m. to 10 p.m. for, I think it was 2 or 3 weeks. And when I say we, I really mean the helpers in my lab, the undergraduate summer employees, did the brunt of this sampling. Every 2 hours, somebody would go out from sunup to dusk and count the number of bees that they saw while walking through the field, and also check these traps for what was caught. And we're still analyzing these data, but the general upshot of this study is that most of the honeybees that we counted in this study were found between the hours of 11 a.m. and 4 p.m. So the third study that I'm going to touch on has to do with neonicotinoid seed treatments in soybean. And this is those of you who follow pollinators, this is kind of a neonicotinoid insecticide seed treatments is a hot button issue with regard to pollinators, because studies have shown that the dust which flies off of the seeds during planting can kill pollinators, can kill bees. So just a little bit of background on neonicotinoid seed treatments. This is a type of insecticide. Often it's bundled with certain fungicides and applied to the seed coat before planting. And then the insecticide is so it's plastered onto the seed coat, and then as the plant is germinating, the plant takes up the insecticide into its tissue, and this is to target insect pests that would be feeding on that tissue. And the 2 most commonly used neonicotinoid seed treatments in soybean are imidacloprid and thiamethoxam. So the purpose of this study, it wasn't really designed as a pollinator study. It was designed as a study to look at pest management from a pest management perspective. How long does that active ingredient appear in new plant tissue? In particular, how long is it providing protection against the pests that it's intended to control? And if you think about the amount of insecticide that's on a seed, an individual seed, that's probably not going to be a lot of insecticide. There's only a finite amount. The plant is not able to manufacture new insecticide. It only has the amount that goes into the seed. So how long does that active ingredient get incorporated into the growing plant tissue? Because that's going to determine how long it's providing pest management. And this is the part that has to do with pollinators. We also looked at whether the active ingredient of thiamethoxam is found in soybean pollen. So we were looking at the concentration of active ingredient in new plant tissue as the soybeans grew, but we also analyzed in pollen to see if it was in something that pollinators would be exposed to. This was a multi-state study. It was led by Christian Krupke at Purdue University. We had 8 locations in 7 states. We conducted it over 2 years, and I was involved in this study actually in a previous life when I worked for 10 years before coming to Ohio. I worked for 10 years as a faculty member at South Dakota State University, so I participated in this study when I lived in South Dakota. Now, if you're interested in all the details of this study, you can access it on this link here on the PDF that's been made available to you. If you bring that up, you'll be able to click on this link and get to the full publication of the study. But I'm just going to hit a couple of points of interest very quickly. First of all, good news, we did not find any of the neonicotinoid insecticide, any of the thiamethoxam in soybean pollen in any of our treatments. So by that time of year, basically the insecticide was long gone and it wasn't in the pollen, and so the pollinators were not being exposed to it by that route. So probably the main source of exposure to these compounds happens probably during planting. Now this is the thiamethoxam concentration in new soybean vegetation, which was what we were trying to see, how long is it available as an insecticide. This is the days after planting, and this is the thiamethoxam concentration in new soybean vegetation. And there's a lot of the thiamethoxam at first, but by basically 2.5 weeks after planting, we've bottomed out and we have none of the insecticide in the plant tissue. So during this whole period after the first basically 2.5 weeks, 3 weeks maybe, we have essentially no product available for pest management in soybean. And this is relevant because first of all, very relevant that the neonic is not being transmitted to bees in soybean, but also relevant for soybean farmers to know that this active ingredient fades away very quickly, and so it only has a very limited window of pest management. And I think at this point all of the field crop extension entomologists in the Midwest have now adopted the advice that at least in soybean, and this is different in different crops, but we're talking about soybean, that we don't recommend automatically using a neonicotinoid seed treatment in soybean because most of the pests that occur in soybean occur later than that window of 2.5 to 3 weeks after planting. There are some exceptions to this. There are a few pests that are particularly early season in situations that if and when they might be a problem. These products might still have value. For example, certain locations, if they routinely have problems with early season bean leaf beetle, or in cases where there are early season soil pests of the seeds and seedlings, for example wireworms. And there are certain situations where that's more likely to be the case, particularly if soybeans are being planted after CRP or after pasture in that first year. Also, another caveat, they performed a similar, they performed some work on the value of neonicotinoid seed treatments for pest management in the southern U.S., mainly in this area here, and they got different results. They have different pest complexes, so the results I'm talking about in terms of efficacy for pest management, really I'm talking about the Midwestern region where this study was performed. So I hope, we're nearing the end of the presentation, but I hope that through these 3 studies I've been able to show you that even though soybeans do not require pollinators for their pollination, still soybeans are an important part of the pollinator story. I would like to thank all of the funding organizations that help support this and related work, and I would also like to thank you for your attention this morning.
Great, thanks, Kelley. Thanks for the presentation and for, I think, shedding a light. There are a lot of questions out there among beekeepers and bee fans about the neonicotinoids, and that's an important piece to help us understand. Folks, if you have any questions, feel free to type those into the chat pod and we'll take a few. Kelley, you were mentioning that when you were surveying the soybeans in different fields, you had, of course, different kinds of soybeans. But in general, how long would you say that a certain soybean cultivar is in bloom? Is that like a 10-day period, or how long are they blooming?
Yeah, I would say 10 days, 2 weeks at most. And then there's a difference between determinate and indeterminate soybeans, and so some soybean varieties will just always have some blooms going. But most of the varieties that we see in Ohio are only have a particular bloom period of bloom of no more than, I would say, a couple of weeks.
Okay, great. Alan asks, what would be the average yield gain in percentage in soybeans with honeybees helping versus not?
Well, that's a really good question, Alan, and that's something that we don't have a lot of information about yet. And in fact, I'm involved in a study that's going to start next year where we try to pin that down a little bit better, like what really is the yield advantage for having a lot of honeybees. Right now we just have some preliminary evidence that you can get up to a couple few bushels of yield increase, which is, when you multiply that across all your soybean acreage, turns into real money. But the data are really too preliminary at this point for me to answer that well. But I think it's a very important question and something that we are planning to work on, because if we can demonstrate to soybean farmers the value that these are providing to a crop, and for a long time people ignored pollinators in soybean. We knew they were there. We knew that soybean pollen shows up in honey, so they must be visiting it. But not a lot of research was done because soybean is self-pollinating. So this idea and this preliminary evidence that maybe they're doing something is agronomically very interesting and potentially important information that where people will be doing more work on.
Hey, there's a question about the different colored bowls that you use. Did you see any differences in the bees that were visiting those different colors?
I thought we would, because there's this whole literature on, the colors and which colors to use, and there's some conflicting evidence, and we actually found, so we only had the 3 color bowls in the study this summer where we were looking specifically at the honeybees, but we also recorded bumblebees, and it seemed like they were coming to all the bowls actually. So I think that a much better study could be looked at with the color thing, but we were just, and we had the best luck really just walking through the field and looking rather than waiting for somebody to commit suicide in one of those bowls. So the bowl thing for the time of day study, where we had such limited sampling windows, was not the best sampling tool this summer. Really, looking was the best tool this summer.
Okay, there's a question as to whether honeybees are collecting both pollen and nectar from soybeans.
That is an excellent question. I do not know. I know that they're getting...
Go ahead.
Denise, go ahead.
I was just going to mention, I know that Reed Johnson, who you mentioned earlier, a faculty member in our entomology department, has done some work in having Ohio beekeepers bring honey samples in, and then they've been looking through those honey samples for soybean pollen. So he has been, and I don't know the results of his data collection, but has been finding that soybean pollen in the honey.
Yes, and he's told me the same thing, that certainly they obviously are visiting the fields since the pollen shows up, but I don't know for them the relative value of the pollen and the nectar. That's a really good question.
Hey, Janice asked about the dust that's given off when seed is planted and how much of a problem this is. Maybe you can just give a few words to that.
Yeah, so this is something that's a point of a lot of active research, and it all began a few years ago when Dr. Christian Krupke at Purdue University was the first really to look at this question and show that the dust that comes off at planting can travel. It can travel in the breeze. It can hit plants that are weeds, for example, surrounding the field, and that it has the potential to kill pollinators around the field. And this is became a very kind of big news sort of thing. And a lot of it depends on the product, and there's a whole science to the formulation of the seed coat treatment, and different companies have different formulations, and also a lot of it has to do with talcum powder that is used as a lubricant when the seeds are being planted. A lubricant is added to the seeds to make everything flow through the planter better. And it has also been shown that this lubricant powder, talc, can pick up, and there are other types of lubricant powder, but that they can pick up some of the insecticide from the seed coat. And so the answer is it's not simple how much dust is given off. It really varies a lot. But I can tell you that, that agricultural companies have started to do a lot of research and development on formulations of coating the seeds that will be less prone to giving off the dust. For example, like an idea sort of like the candy coating on an M&M, some type of application technology to reduce that dust. So there's a lot of interest in developing those technologies.
Hey, Kelley, Alicia has a question about GMO soy products, and I know it's not really what we're talking about today, but if you want to, I'm sure this comes up a lot when you're presenting both to the public and to growers. Can you give a little summary of what's, what those varieties are and how they're used in soybeans?
Okay, so most, in fact, in the U.S., all of the genetically modified soybeans are modified not for pest management. There's no Bt soybeans in this country. There are in other countries, but modified to be herbicide resistant. So, for example, Roundup Ready soybeans are genetically modified so that the herbicide Roundup doesn't kill them, but will kill the weeds, so the farmer can spray the field, not kill the soybeans, but kill the weeds. So there is absolutely no evidence that that genetic modification affects the food quality for things like tofu and soy milk. You can't find any trace of it in those products. So just in terms of health benefit, there's really no evidence that soy milk from GMO versus non-GMO soybeans is any different. Now, if you are interested in things like supporting supporting agricultural field management that reduces the use of herbicides, and so you're interested in sustainability issues where less herbicide, other weed control methods other than a heavy reliance on herbicides is used, that there might be some value that way to choosing non-GMO products. But from a health point of view, nothing that I've read in, and I have followed this issue in field crops about the effects or non-effects of GMO in human health. And everything that I've read that's a scientific study, properly conducted, suggests that these GMO soybean products, there's no health detriment on them.
Okay, Beth wonders, when you were collecting honeybee counts this summer, did you find a lot of solitary bees in those counts, or were you focusing really on the honeybees?
We were really focusing on the honeybees, and a lot of these other bee species are sort of small and subtle, particularly the sweat bees are not easy to always see. And so we were really bumblebees, big, obvious, and we counted those when we saw them. But the solitary bees are a little less flashy, and so and we were using a lot of non-trained people, by which I mean undergraduate workers who were hired to help us during the summer, and so we couldn't really expect them to be doing full-on bee identification in the field. So our focus really was on the honeybees. Although when I was out doing my share of the counts, I did, see some of the other things, but mainly, you get into a mode where you're really just trying to get through it, and you're looking at the honeybees.
Okay, there were questions about whether you noticed varietal differences in the soybeans, and also whether weather played a part in your results.
So because we were sampling in different locations, we couldn't separate variety from location. So like if variety A was in Ashtabula County and variety B was in Licking County, and we saw differences in the bees. We couldn't say whether that's because one's in Ashtabula and one's in Licking, or because one's in this soybean and one is in that variety. So our study was not really set up to be able to tease that out. But in terms of weather, definitely we can see differences. For example, one of the graphs had a sampling week where there was a really lower counts average-wise for that week than the weeks around it, for no obvious reason from the graph, but it happened that that was a very cold and rainy week through much of the region. And so in that kind of weather, where it's kind of cold or rainy or windy, definitely we would see less activity in our catches in the soybean fields. And also in the time of day study, if we were walking through and it was a kind of a cold, cloudy, rainy day, we would definitely see less going on. Good question.